Morning, everyone. Hope everyone had a restful night. I just had a few announcements. The new ones are in blue. The posters are scheduled for 8 to 3 today. Many of them are up already. Hopefully they'll all be up soon. So they will be available until just before 3, in which they actually have to be taken down so they can get ready for the dinner, which we'll have in the same space in the Great Hall. But we do have an in-person session from 1:30 to 2:30, and then after that session, we'll have the posters down. Just also wanted to announce that they've fixed the time and date for the next FANG workshop, which will be at PAG, Plant Animal Genome Meetings in San Diego on January 11th, from 6 to 8:30 p.m. It's a Monday. Any other announcements? No? Okay, I'll switch the slides. And you
Okay, so we have two distinguished guests to welcome us this morning. First up is Dr. Sunny Ramaswamy from USDA NIFA.
All right. Well, thank you very much, and thanks, Chris, for inviting me to share it with my. I don't need it. Do I need it? For the online people. Oh, for the online people. My bad. Sorry. So thanks very much, Chris, and Lakshmi as well for having me here, and welcome, Rob. You'll follow me, I guess, and share a few thoughts as well. And Chris was very clear to me, saying that no more than 5 to 8 minutes, I think he said. He must have known that I've been a professor, you know, long-winded professor types, right? Jerry knows a little bit about this too. So seriously, really appreciate actually, Chris took the time to fill me in quite a bit about the workshop itself and the goals and the objectives and things like that too. And so I just wanted to share a couple of words with you and then let Rob speak from NSF's perspective as well. First of all, I mean, I really appreciate all of you taking making the effort to take all the data that we've developed over the last many years in regards to genomics, genomic data, and now trying to understand what that means in regards to the phenome itself. And in my conversation with Chris the other day, I was saying that, you know, the plant people are way ahead of the animal people, at least from my perspective. But by the way, I'm an entomologist, and I did use some molecular tools back in the day when I was a practicing scientist. And now I've been an administrator for a very long time. So, but still, you know, the plant people have been quite a ways ahead. In fact, one of his colleagues from Iowa State University and folks at UC Davis and other places, Patrick Schnabel, some of you might know, they're doing some really cool work in, you know, taking the genomics data and trying to understand what it means. So they've got this project called Genomes to Field, for example. And something that you might want to consider doing collaboratively, collectively, and I'm going to come to that just towards the end of my few comments that I want to make as well in regards to funding itself. This is critically important for us. All the data that we've had for all these years, what do we make of it, and how does that contribute to, ultimately, for us, the National Institute of Food and Agriculture, is how to ensure that farmers and livestock producers are going to be impacted in a positive manner. And some of the kind of work that I know personally that Jerry and his group, they do in the Midwest is really critically important for the kind of things that enables farmers to achieve what they need to achieve as well. That's something that we need to bear in mind. It is user-inspired science that we're deeply interested in, that we support as well. And I know that those of you that work with livestock animals at various land-grant universities and other places know about this as well. So that's the first thing to bear in mind. So we're deeply interested in providing the support that you need for that kind of work. And so we've had multiple in the very recent past, again, going back to the plants area, for a very long time, NIFA and NSF have partnered for a very long time in providing funding to the plant community. And in the animal community, it's taking a few baby steps yet. And Mark Morando is sitting in the back there, and he and others of our colleagues, Lakshmi and Peter Johnson and others, work with NSF and NIH and the British Biotech Research and Science Council, BBSRC, and the European Union, by the way, in trying to figure out how best to provide the resources necessary, you know, essentially crowdsourcing the intellectual resources, but seriously into crowdsourcing the funding as well, the monetary resources as well. And we're committed to continuing to work with the various interested parties, particularly NSF. And again, going back to the plants area, Ann Litchens Park is one of our staff members, scientific staff members in NIFA, and she is going to be putting out joint RFAs in the plant microbe interactions area, for example. And when we do these collaborative RFAs, the intent is to, those of you that have received funding from the Dual Use, Dual Benefits program or the Ecology and Evolution of Infectious Diseases program, no, we convene these RFAs together, we convene the panels together, and we make decisions. And then, we parse the projects to be funded by the different agencies as well. And it really allows us to leverage the resources that we've got. And the animals area, we made a commitment to continue to increase the investment that we make. And I guarantee you in the last few years, we have indeed increased the investment. But our commitment is to continue to provide additional resources for the animals area as well. So that's something that we're committed to, and look forward to working with Lakshmi and Mark and the others in the agency, and particularly with Rob and the others in NSF as well, on continuing to focus on our collaborative efforts in supporting the work that you do. The last thing, going back to the Genomes to Field project that Patrick Schnabel and others have developed, Iowa State University and others are involved in this, multiple others are involved. They brought together the various commodity groups, the corn folks and the soybean folks and others. And within NIFA's construct of funding that we provide, we have a new avatar called Centers of Excellence. Congress told us as part of the Farm Bill that we have to provide funding through our Agriculture and Food Research Initiative, our flagship program, to create centers of excellence. And you might want to collectively think of how you might create center or centers of excellence to address this, functional annotation of animal genomes and things like that. And to bring in your interested stakeholders, the commodity groups, whether it's the Cattle and Beef Association or the pork people. I know the pork people are really interested in the kind of effort that you're involved in, but bringing in collaboratively all of these interested parties and putting together a significant ask. You could come to directly to NIFA, or maybe it's collaboratively with NSF as well. Something for you to consider, and it's on our website. In fact, maybe Lakshmi and Mark the rest of the day, and Parag Chitnis is going to be here later on. I'll tell you a little bit more about the Centers of Excellence idea itself. So with that, I want to go ahead and stop here, and thanks very much again, and I wish you all the best. Look forward to seeing all the wonderful stuff that you're going to be doing here over the next many, many years. Again, good luck to all of you.
We also have Dr. Rob Miller, who's the acting division chair in the Directorate for Biological Sciences in the Integrated Organismal Systems.
Okay, thank you very much. Since I'm a relatively new face around here, I should introduce myself a bit more. I'm now currently the acting division director in iOS, and I've actually been involved, though, for a long time with some of the programs you just heard about, symbiosis, defense and self-recognition, and have been helping to work on lately this sort of joint solicitation that's going on between NSF and NIFA on plant genomic or plant microbe interactions. I wholeheartedly agree that having been, and I'm an immunologist who works on animals, having been in, so I'm coming at it from that perspective too. I agree with everything you just heard, that sort of the plant community has been sort of moving ahead in this area, and I'm excited by meetings such as this one and being part of this kind of group to see this sort of movement now on the animal side of things. I've been involved, when I'm not wearing my administrative hat at NSF, I'm an immunologist who for 20 years has been involved in comparative genomics as it relates to the immune system, and have been faced with the reality that a lot of you have and a lot of your colleagues of the fact that we have all these data now that we're generating, and we have to translate it over into its relationships to phenotype. And this is becoming a big area of interest in my division and across the biology directorate at NSF. So I'll, to keep us on track here, I'll keep my comments short. You'll hear more about a lot of what we're talking about from the agency perspective later this morning in the session. And welcome to this. I'm very excited to be here. This is sort of the kind of meeting that I've been looking forward to for a while. This is my first venture into it, and I'm excited to be here. Thank you.
Okay, and next up we have Dr. Elisabetta Joffre, who's here from INRA, the French National Agricultural Research Institute, and she's also a co-chair of the Animal Samples and Assays Committee for the FANG project.
Thank you. Can you hear me? Yes. Okay. Yep. Good morning. So I'm here to give you an overview of the status of where we are with the FANG and the data production. So the contents of this talk, I will take back the context, which is essentially what we outlined in the white paper, which Chris already anticipated yesterday. I will report the current progress of the Animals Samples Assays Committee, the so-called ASA committee. I will give examples of FANG samples and data collections so far. And from here, it's very important that these PN people who are interested to join, because there are several items which are emerging and important for discussions and further. We give examples, I mean, 2 pilot FANG projects and a very short report on those. The FANG white paper, essentially we put forward the unique value of domesticated animal species, and essentially for the wealth of genetic and phenotypic diversity, which has been shaped by natural plus artificial selection. And the FANG project that we put forward was essentially divided into 2 phases. In the first phase, which is where we are now, the idea is to obtain a biological reference for main species for which at least there is at present a sufficiently annotated genome. And then to start the so-called genotype to phenotype phase, where the interesting stuff is tackled. So in this case, it means to expand, to collect the different developmental stages and the different physiological and environmental conditions, to collect more breeds, more populations. And this is important also because it's the phase where we can involve industry and commercial populations. And of course, there is also the issue of additional species. Think, for example, of how many aquacultural species exist, which in fact they don't have yet a good annotated genomes, but for which things are advancing pretty fast. And concerning specifically biological targets and assays for this phase of data creation, we indicated guidelines in the white paper. The first one is that members are committed to collecting, storing, and sharing samples for this data collection, the first phase. These collections, trying to keep them simple, address in general a single developmental stage, which is adult, which is score like a quote, because adult stages, I will say later, could mean different things. There is a focus on tissues and primary cells and not on cell lines, which is actually taking what is the latest phase of development of several ENCODE projects, like the mice project. They made use a lot, as also John Stan yesterday said, of tissues and primary cells. And to use well-established assays, which essentially work, let's say, easily and across species, so they don't need really extensive optimization. The ASA committee has for aim to propose, develop, and standardize animal tissue collection, protocols, storage practices, and assay protocols for the FANG community, for the so-called pilot projects that we are carrying out now. And of course, the general idea is that we should foster collaborations, avoid redundancies, and enhance synergies whenever possible. At present, we have 52 members, and the works done so far for collections is outlined in poster number 4. We have collection at present from 6 species, which are in the picture. So you have pig, you have sheep, you have cattle, you have goat, horse, and chicken. What we have been doing has been first adapt and optimizing protocols for assays on tissues, which was not so straightforward as maybe some people expected, because working on tissues is still quite challenging compared to cell lines. At present online, we only uploaded probably the simplest protocol, which is for RNA extractions suited for using SNAP frozen or RNA later preserved tissues, which provide RNAs of qualities which is okay for RNA-seq and small RNA-seq. In pipeline, we have various things. Procedures, for example, for optimal sorting from blood cells, and as well for long-term storage, in this case, compatible for all possible assays. ChIP-seq procedures for histone marks, essentially, for at least some frozen tissues, and Hi-C on dissociated liver cells. Hi-C is a technique we carry out in IRA now, which I will say later better. This is an example, so you don't have to really focus on details of this table. This is the collection that we have accumulated right now for chicken and for cattle. So we have one mammal and one bird. And you have different columns. So you have line breed, so a type of population, let's say. The developmental stage, we should be adult, but kind of differentiated. Tissues and or cells collected, which country of origin, the number of tissues, flesh, and which assays are conducted. And one first thing which emerges from this table is the genetic diversity. So here there is not a minimization of genetic diversity. In fact, there are different breeds and different lines and even crosses. Second, you see that there is a wide range of definition of adult stage, depending on when the sampling was done. And physiological states may also differ between these animals, because, for example, if you collect animals from a commercial facility or from an experimental facility, you may work in quite different conditions. And in general, also, there is to take into account the technical score heterogeneity that you can have in how you sample a given tissue and in how many aliquots you produce, for example. And this kind of issues, one way that we thought for organizing the collections is to use biosamples, which is a database of sample descriptors realized at MBL EBI in UK. And in the next talk by Laura Clark, you will hear more about the committee, the metadata and data sharing committee, which is proposing actually this tool. And what is the idea is that we can catalog, organize and catalog all the FANG samples, which will be automatically connected to all metadata and associated data sets. And once this will be done and there will be a critical mass of data sets, it will be easier to assess common feature across species, and also importantly, I think, to measure this heterogeneity, technical and biological heterogeneity, which can arise from a technical point of view, for example, among different portions or aliquots of the same tissue, of the same organ. And for this collaboration with this committee, the ASA committee is helping to define the minimum requirements, which suitable ontologies we should use. And in this way, we will proceed also to choose is using examples, metadata, and so on. So this tool, I think that helps us a lot, because it means that all this heterogeneity of current collections is not just a challenge, but it's a nice opportunity also to possibly anticipate the next FANG phase. by John Stam, 4 histone marks, and the CTCF transcription factor binding site. And at present, this is the slide that Weiju sent me, we are advancing, trying to complete the first picture, especially for the chickens, and they are quite advanced for RNA-seq, and they have quite a good coverage also for the soft tissues, essentially for DNA-seq, and 2 histone marks. Project 2 is called Phage Encode. It's funded in France by INRA intramural grants, essentially by an INRA meta program, which is interestingly is a meta program to foster research on genomic selection. So they identify quite early the importance of annotation of genomes for the perspective of genomic selection. In this project, we collect the old tissues from 4 species. We added the goat compared to the American study. And while all tissues are stored, we targeted only very few tissues for the first phase. And this was essentially due to limited funding. And the choice was also dictated by the need to benefit the parallel ongoing research, which is going on at the different FANG institutions, different INRA institutions which are participating. Note also that it is already in collaboration with the other core FANG partners in Europe. The collections, it was a huge work that was to set up, and it's carried out in collaboration with the National Infrastructure of Biological Resource Center for Domestic Animals, which is a huge national infrastructure in France, which actually is an interface between us in FANG, in Phage Encode, and experimental animal facilities of INRA. So the idea was to collect samples also from populations which are staying, so that they can be sampled again, and also the animals we use, the sperms is preserved so that we can produce progenies. What we achieved, it's the biorepository of 4 species, where in our case we use 4 biological replicates, 2 males and 2 females. We snap freeze several aliquots of all the tissues. We preserve blood cells because we have a specific interest to blood cells, which for the moment we sort only for these 2 types, but then PBNCs will be used for sorting additional types. And by end 2015, we hope we will have optimized Hi-C and ATAC-seq. Didn't say before, I jumped the line, but we invested energies to try to optimize these 2 assays. Why these 2 assays? For several reasons, but let's say this is an emerging method which allows to really map interaction between long distal elements and, for example, target genes. It's a bit difficult to carry out, but it's also very, very powerful. So we thought it was worth it, and there was research ongoing on that already. And ATAC-seq represents a method for characterizing chromatin to detect which accessible regions are there. So it's a kind of simplification of DNA-seq, and it's a much simplified because maybe it's not as powerful as DNA-seq, but you need just 50,000 cells to carry it out, and it's extremely simple as procedures for library preparations. And by 2016, we will start we will make all the sequencing around January and start bioinformatic analysis. All of sequencing runs will be done together also to avoid batch effects. And there is a poster to give you an outlook, a view of how sampling was performed in 2 of the species. And then I think I'm over with presentation. The invitation is really to join us for breakout 1, data creation, this PM, where the idea is to take again these things that I labeled in previous presentation and discuss the current challenges in standardizing sample collections and assays, which are due to the specific complexities of domesticated animals, and also explore options, because this is the most important thing, to really organize ourselves and to choose the direction to go in a very coordinated way from now on. Thank you very much.
Very much, Elisabetta. Does anyone have any questions?
Yeah.
Yep.
Yes, I have a comment. The comment is that we just sort of need to be aware that the reference assemblies that we're dealing with. Sorry. My comment is just simply that we need to be aware that the reference assemblies that we're working with are draft assemblies. And are problematic in most cases. I mean, as soon as you see data that say 90% of your RNA-seq reads map, you have to ask the question, what happened to the other 10%? And the majority of those represent exons that are not present within the reference assembly. In cattle, it's worse than that. We never map more than about high 80s, 80, 88, 89. And we've actually we have a paper submitted that characterizes what's missing in the UMD 3.1 reference. So they're evolving, and in fact, there are efforts underway to generate PacBio assemblies for some of the species, certainly in cattle. And when we get those done and those annotated, a lot of this work is going to need to be repeated if it's all based upon the current reference assemblies.
Yes, well, first the note, I don't think that the slide referred to percentage of mapped reads. I think it referred to percentage of assays which were completed, right? Yeah, so it's not it's not a percentage of mapped reads. This is different. So maybe the mapped reads are more, but of course you are right, because there is a problem of genome annotation. And actually many of these assays actually should help also the assemblies. Like HiC, for example, is a technology which is extremely interesting for filling, for the scaffolding, yes. So, I mean, it's a doubt, that's
I just wanted to make another maybe comment on those lines, and is that, I mean, respecting to gathering the information in terms of samples and sequences from this assay, it doesn't matter, the assembly doesn't influence the information you gather. So this is all sequencing based, so the sequence, biological sequence will not change because our assemblies change. So that the analysis will be, I mean, an iterative process that can be redone and maybe re-evaluated.
Yeah, I think that, I mean, the genomes will never be fully annotated in one. I mean, it's a continuous process by definition. And so in a continuous process, you continue to rerun, not to rerun things completely, but to gain insights.
There are empty seats at the front, so if anyone wants to move forward, please do. Thank you.
Yeah, I think wonderful job. So I want to make some comments. So you mentioned about some progress from chicken and also the dairy stuff. Actually, we also made some progress about the, for example, chicken, but not only from the F1. We made some progress in parent line 63 and 72, and some data set already listened to NCBI. And so this one. Another thing is, Jerry and I, we also collaborated some project. Actually, we worked this for years. For Angus beef, I think maybe this is very, very unique. We collect some more than 15 samples and from individual. So that's grass-fed action. I mentioned about that. So this sample are already ready there. Some data set we already generate. So if we want analysis that, we are willing to contribute this data set to our community.
Of course, yes. And my recommendation is uploading biosamples as soon as all the terms and methodologies are defined. I mean, that would be the best tool we have, I think, to really have everything organized and not redundant.
Jerry, another one?
Yeah, go on. You know you can do it. So you went fairly quickly through the list of tissues, but I didn't notice any tissues there that were immunological. And I just want to make the comment that the USDA has funded a respiratory disease project here in the US. And in that project, a fairly large number of animals have been challenged with specific pathogens that cause respiratory disease, and then those animals were slaughtered and a lot of tissues were harvested from those animals. And in fact, we've published some RNA-seq work based upon the bronchial lymph node. We've completed RNA-seq on another 4 or 5 tissues from those animals. And those tissue samples are available, and the RNA-seq data has already been paid for. And it might be something to consider, given that I didn't see anything in there that was based upon immune function. So they may be very useful.
I didn't go into details, but of course they are. I mean, you have all the tissues which are, I mean, related to immunity, of course. I mean, from lymph nodes and whatever, and the gut in the gut, the gullet, the tissue, in the intestine tissues, and so on. It depends a bit on who is sampling, but for example, there is a project submitted in Australia where they collect 78 tissues from cats. So you have everything actually, and immunological tissues are represented. And the good, interesting thing could be to actually, I mean, at some point to go to see these collections and see how data could be matched and analyzed.
Okay, can you join me in thanking Elisabetta for her talk?
Thank you.
Thank you, and it gives me great pleasure to introduce our next plenary speaker, Christine Wells, who's a reader of immunology in the Infection, Immunity and Inflammation Institute at the University of Glasgow. And she also spends half of her time at the AIBN in Brisbane in Australia. And she's been heavily involved in the FANTOM project, and she's going to give us a perspective, I think, on the data from the FANTOM project. Thank you, Christine.
Thanks very much, Brian. Can you hear me okay? Okay. All right, how are we going now? Great. So I would like to thank the organisers very much for inviting me to come and talk to you today. I'm going to give you a little bit of a mud map on my experience with the FANTOM project, which FANTOM stands for the Functional Annotation of the Mammalian Genome. So very, very much aligned to the goals that FANG have lined up. And a mud map in Australian terms are essentially these crude drawings in the sand. And the idea is that you want to let people know a route where you want to go, but you may not find that giving them a direct set of roads or paths is particularly useful. Frequently what you want to give them is just some landmarks to look for and let them make their own roads there. And I think that this is the message that I'm hoping to give you today, is that I'll let you know what worked for us without necessarily trying to be prescriptive about the direction that you should be going in. So the first point that I want to make is that we often apologise as genome biologists that we're stamp collectors. And the truth is that's never been the motivation for a genomics project. Genomics projects have always been grounded in this question of phenotype. And I think it's very important that we don't let detractors of large genomic projects dictate our agenda in this. So clearly there are issues with understanding function when we collect a large amount of data, but it's important to realise that this genome to phenome gap has always been at the heart of these projects. And even before the mouse and human genome projects were published, the annotation of transcriptomes were really important in building the structure and architecture and annotation of those genomes. So clearly, having a good genome project does mean that you need to be collecting the right kind of data. And one of the genomics challenges, I think, is that we are still very much embedded in the idea that phenotypes are entirely protein driven. But of course, expressivity of phenotype and penetrance of phenotype are extremely important concepts that we learnt about as geneticists well before genome days, and we mustn't lose sight of those lessons. So many of the layers of information that we're pulling together may not be driving the direct gene to protein question that we're entrained to test now, but it may actually be important for the expressivity and penetrance of the traits that we're interested in. And if this is the case, then some of the phenotypic assays that we want to employ need to reflect this question rather than just a completely deterministic view of the way the genome works. And of course, the great thing about including information on the epigenome is that we've got capacity to start modelling the impact of environment on expressivity of these traits. This is the reason that we attract so much criticism in terms of the potential for reward of these so-called stamp collecting projects. The fact that you can gather a whole pile of data and clearly that is not representative of biology. But this also ignores the fact that those of us who are generating the data are firmly grounded in strong biological questions and frequently have trained in our careers to address a particular aspect of that biology, and these are tools that we are bringing to address those questions. And I'd also like to point out that many of the detractors of genomic projects are also very quick to dismiss some of the most important discoveries from these projects, such as small and non-coding RNAs. And even now there is a large debate about the functional relevance of many of these regulatory regions that have been described by ENCODE, many of the transcripts that have been described by FANTOM, and clearly just compiling these is not going to be sufficient to say, yes, these have a purpose, but ignoring them is also ignoring things which are obviously fundamentally important to the construction of the organisms that we're very interested in. So it's my opinion that to really make the most of large-scale genome projects, we need to be taking a systems biology approach. And this means that we need to be thinking very clearly about these principles of systems biology, such as emergence, which is the idea that by integrating a large amount of data, we're going to find insight which is not visible from a single study alone. And I'm going to give you some examples of this from the FANTOM projects. This question of modularity, which I think is absolutely critical to a genome annotation, and that is that we're moving away from examining individual genes and starting to examine the molecular networks that drive and sustain phenotypes that we're interested in, and these include both protein coding and non-coding elements. And this question of robustness, which I think is the hardest question to address, but probably the most important, and one of the reasons that I'm so excited by this FANG consortium, because from my perspective, this question of robustness in molecular networks cannot be addressed from a single species perspective. And frequently, if we are single-mindedly looking at the sequence to protein content, then we're not really considering all of the elements which buffer a network from small genetic and environmental perturbation. And really, if the conservation around these networks is in function and not sequence, then we need to have information from a large number of genomes to be really understanding how these buffering elements work. So there's huge excitement from my perspective as a genome biologist in understanding how these modules are regulated across a large number of animal species. And I think that this will be a very important resource for the entire community, as well as for the agricultural community for this reason. And of course, you're already talking about the things that need to be resourced in this area. You know, we had a very good talk from Elizabeth on how important it is to generate quality data, and a lot of that does come down to setting the standards for what you want to have. Equally, tools for visualizing, integrating and sharing this data are very important, and this is not a trivial aspect at all. It needs to be resourced, and it needs to be resourced not just for the bioinformaticians who are comfortable working with data, but it must be resourced for the biologists who perhaps have poor bioinformatics capacity but deep biological insight. And this is a much harder thing to do because their needs are, it needs to be fast, it needs to be direct, and it needs to be intuitive. And a genome browser sometimes is not the best way of looking at modules or networks. And I don't have a solution for you here except to acknowledge that it's a hard problem. And I think that this again comes down to very good collaborations between bioinformaticians, biostatisticians and biologists. And I'm very heartened to hear that many of you already have good established bioinformatics collaborations in place, and I think this will be a key to your success. So we saw this slide last night, and it's certainly one that is beloved of genome biologists. And I think Conrad Waddington had enormous contributions to make in this field of genome annotation and genome regulation. But I'm not putting it up here to talk to you about the epigenetic landscape at all. I'm putting it up here to talk to you about the canalization of traits. And again, this comes back to this concept of penetrance and expressivity of traits. Because canalization is not just about getting this ball down the hill and restricting its path. It's also about providing for phenotypic variation within a population, even if that population has got a fairly narrow genotypic background. Because biological variation is one of the key outcomes from a fit genome. And that doesn't just come back to sequence polymorphism, but also comes back to the diversification of genome products. And some of this will be determined by the underlying sequence and the proteins that it encodes, and some of it will be stochastic. And including these stochastic processes in our models is hard, but it's really important. And when we consider a lot of the modularity that might be arising from these genomes, then we might consider that stochastic does not mean random. What it means is constrained variation that's controlled, and that many of the elements we're looking at may not be strictly additive in terms of the traits that we're driving, because what their role is to influence the probability of that from the scale of highly probable to not probable at all. And of course, when you're talking about canalization in a classic Waddington perspective, then the developmental timescale is extremely important, because molecular networks that are important at one stage in development and tightly constrained may not be important at a later time point, and therefore their regulation is going to be much less important to the expression of that trait. So getting the tissue context and the developmental context right is important up front. So let me just walk you through why I'm a huge fan of Atlas projects. I've been involved in FANTOM since the beginning, and I'm hugely enthusiastic for the next round of FANTOM 6, which has just kicked off. So the FANTOM structure is one that is a little bit dissimilar to the standard ENCODE or Human Genome Project structures, because it's been entirely funded and driven from a single research institute in Japan, and that's the Recon Genome Sciences group. And these researchers have really driven the technology development, as provided the computational infrastructure and coordinated the project. And the advantage of this to us is that the data generation has been centralised and the data management has been centralised. And that means that it's extremely reliable in terms of knowing what it is that we're looking at. Now, clearly that's not the model that you will be adopting because you have so many different groups involved. But thinking about data harmonisation is going to be an important part of the success of your project. And sitting either side of this core group, you have the bioinformaticians, biostatisticians and computational biologists. And there's a real sandpit model here, which is extremely successful because what has arisen from FANTOM has not just been a catalogue or an atlas of RNA expression across different tissues, but also a large number of new tools and approaches to handling this data as the technology has developed. And equally important, but perhaps interacting in a very different way, are the sample providers. And here I think there are lessons for the FANG community, because these individuals generally are biologists with deep domain knowledge in one particular area of biology, and they gain a lot of advantage in being part of FANTOM because in addition to having a very good deep data set in their own particular biology, they have access to the larger embargoed data set. And this comparative power of the large atlas is very important. But this group is difficult to manage from a consortium perspective for many reasons. And I think the main reason here is that without embedded bioinformatics, the sample providers have almost universally struggled to find the data that's relevant, let alone use it. And part of this just comes down to visualization. And FANTOM and RECON in particular have done some very good jobs at building genome viewers and wikis to share data. But actually, these are built by bioinformaticians for bioinformaticians, and they're often just overly complicated. And there needs to be a point where you ask your community, what is it that you need to see? And just give them that. So having a good, frank communication with the people who are going to make best use of the data, rather than just expecting that the bioinformaticians should tell you what you need to know, is an important aspect of data management. The other problem that many biological investigators have had in FANTOM is that they simply have come into the project with unrealistic expectations of what will be delivered to them. And this is particularly the case in terms of bioinformatics support. And so, although RECON is very good at providing basic bioinformatics support to the biologists, generic bioinformatics just doesn't work. Service bioinformatics is an abject failure. And this consortium should be no different, because the truth is, to get a question to a biological answer, it needs to be a strong engagement between the biologist and the bioinformatician. And so this proactive engagement actually needs to be proactively managed, and proactively managed by the biologists as much as the bioinformatics community. And many people are very naive about coming into this. So setting out expectations early in your consortium can save a lot of grief. The reason that you want to have lots of diverse biology here is because it's these people who are actually going to frame the biological context, which is enormously important when you're moving away from the stamp collecting criticism into actually driving very deep insight into the data and its application to a particular system. And in particular, it's the biologist who's going to provide you with that strongest link to phenotype, which is so important, because at the end of the day, all of the genomics data that we collate is only correlative, and only correlative with the quality of the phenotype that you've managed to accrue. So the analysis and focus in biologists is often on their favourite gene, which can be frustrating to a bioinformatician. It feels like cherry picking, or on a gene network or pathway that they already know about. But you know, there's a good reason for this. They've got the tools to investigate it. They already know something about these genes in their biology. And so I say, have at it, let them go for it. You know, the bioinformatics and computational biology community This has been a really enjoyable aspect for me coming into the FANTOM community as a non-bioinformatician and building bioinformatics capacity in my own group as a result of this interaction, and now considering myself to have strong bioinformatics credibility. So I've really learnt my bioinformatics in this sandpit, and it's been fantastic. And this is a really wonderful opportunity for your students and early career researchers to gain access to a range of different bioinformatics approaches, not just specialisation in, for example, SNP analysis or EQTLs, but also in actually understanding quite different and strange data structures. So you need to have some agreed terms of mutual respect when you bring a sandpit approach into data. What I mean by that is, again, expectation management. So the biologists need to know that the bioinformaticians involved in the project are not there to service them, but it's not a "you're working for me" scenario, and people can forget that when they're very motivated on a single outcome. But also, actually, the bioinformaticians need to know that they don't have ownership over data that has come from a large backstory and may have had a lot of blood, sweat, tears and history from the biologist's perspective as well. So this mutual respect is really important. It needs to be formally enunciated. In terms of bioinformatics, I feel there is value in redundancy of effort. This is the danger of the sandpit approach. People will approach the same question different ways. As a bioinformatician, you can get very burnt from this because your approach may take a lot of time and effort and not be adopted. But from a consortium perspective, what it does is it gives you a fertile ground for picking the analyses which are really helping you drive insight across the entire data set, and I think that's very valuable. So there is also obviously danger in computational silos, and this is where people will run off and run exactly the same analyses without talking to each other, and obviously you want to avoid that too, so that's a fine balancing act. For bioinformaticians, there is a very good opportunity to form close and productive collaborations with biologists and biological data sets that they may not otherwise have had access to, but you do have this danger of food court bioinformatics. What I mean by that is that the biologist shops around and has a little bit from here, a little bit from here, and that your work as a bioinformatician may not end up in their paper, and your efforts may not be acknowledged in authorship. And this is career suicide for many young bioinformaticians. So again, it comes back to this question of mutual respect. I believe very strongly that bioinformaticians need help framing the right questions in a biological context, so this is a really important collaboration. And I also think that as a consortium, you will experience a big range of computational rigour and quality, and it's important that you set some standards early on in this. We've already discussed the opportunity to form deep domain expertise in specialist data types, and I think this is extremely important. Then there's a third group involved in FANTOM, and that's this strategist group. This is a strategy group that's been drawn in across all of the layers of FANTOM, and the individuals that migrate into this group are the individuals with the broader vision in terms of systems biology or genome biology, and they're interested in developing rules of systems behaviours rather than investigating any single one system. Generally, these are extremely good communicators. They're enthusiastic for science, and that drives the project along. They meet frequently, and they plan and execute the publications, and they recruit people who may not be part of that strategy group into tasks within that publication. So early days of FANTOM, this strategy group was somewhat artificially put together, and one of the things that FANTOM has done very well over 5 iterations is find the talent and allow that talent to come to a fore. So just to give you some specific examples of the output of FANTOM and why I think Atlas projects are so important, this is Yoshihide Hayashizaki, who was the founder of the FANTOM project. And I met him in 1999 when he did a tour of the UK talking about this crazy scheme to build a book with a representative gene from every representative transcript from every gene, full-length cDNA, and he actually printed this book. There are, I think, 12 copies in existence of this book. And he went around the UK and people universally dismissed him. Who is this guy? Why does he think he can do this? You know, the genome's being sequenced. Why do we need an RNA catalogue as well? And actually, I think he was extremely visionary, extremely persistent. And you must remember that the late 90s, early 2000s, where there was a lot of skepticism about the value of the genome project itself, let alone these additional layers of information. But because he was so committed to this, he managed to recruit crazy individuals from around the world as well. There are about 200 of us sitting across different countries with a range of skill sets. And FANTOM did indeed set out to catalogue this representative messenger RNA for every protein coding gene. It built a lot of the computational infrastructure needed for gene discovery and open reading frame annotations. And even today, 2005 and then again in 2014, we had great catalogues of gene expression across different atlases as a result of this vision. And for me, as a young macrophage biologist, this was very exciting because this is a mouse study where I had one mouse strain which was a low responder to endotoxin and a mouse strain which was a high responder to endotoxin. And I was able to print some cDNA arrays from these large full-length cDNA libraries from Recon. And what I discovered is that all of the genes that were well annotated and known in the immune response sat up here as the very abundant and not very interesting in terms of change between time or species. And all of the genes which were very interesting from an immunological perspective were the ones that were transient, and they were also poorly annotated in terms of the genome at that time. So using these transcript-based models, what we learned was that, you know, you can enumerate many members of protein classes, but that actually a lot of transcripts are very transient. But these transient transcripts are often the ones that are most important for phenotype. So I second the suggestion of looking at immune function in your animals, because here you're actually looking at a phenotype which is a very, very important from a biological perspective, but also is going to allow you to find a lot of these transient transcripts which might otherwise be hidden from homeostatic tissue modelling. I think the other lesson from FANTOM is that it really expanded our view of the genome in terms of non-coding RNAs. And FANTOM has continued to push this question of non-coding RNA very seriously. So, for example, it was from the FANTOM projects that we learned that non-coding RNA is the major product from RNA polymerase II, not a minor product. And even though we still have this debate about how much this is phenomenology, sorry for the typo, or a new functional class, and the answer to that still requires a lot of data integration, I think that we're making huge headways in this non-coding world. So back in 2005, 2006, there was the first kind of nomenclature and atlas of widespread sense-antisense transcription in the human genome, in the mouse genome and the human genome. We now know a lot of these RNAs have particular functions. For example, a lot of these bidirectionally expressed RNAs sit in enhancers. And this is the third lesson from Atlas projects, is that when you start opening your eyes to these new classes of molecule, you're actually able to see some new behaviours. And something that we started to observe in a small number of cell lines and primary macrophages, for example, we were able to generalise to a large number of transitioning cell types and actually start to define some new rules. And in this case, what we described was that enhancers, actively transcribed enhancers, are remodelled very quickly following a stimulus. that these then are followed by expression of the transcription factors which dictate the new steady state of the next stage in that cell phenotype, and that finally these late stage genes are the structural genes which dictate the function of those new cells. And this is work that has really been pioneered by Robin Anderson, who was working with Albin Sandelin at the time. Robin's now just started his own group in Copenhagen. And here they were able to show that these actively transcribed enhancers are really cell type specific, that they can map them to a range of different distances to proximal promoters, and that they're very prescriptive in terms of lineage. So the advantage of using CAGE to find these enhancers is that you've got a double bang for your buck. You're getting a molecular map of transcription of the messenger RNA because they're capped, but you're also starting to footprint some of the regulatory regions which overlay very tightly with the more traditional chip-seq methods. And then lastly, I think the importance of Atlas projects is that they really increase the power and potential of molecular phenotypes. They help us find useful surrogates for behaviour of chromatin, and they help us understand complex biology. And what I mean by useful surrogates is that we often rush in to do lots and lots of stuff without really understanding which measurement can substitute for another. I think that the roadmap project was fantastic from that perspective in identifying the minimal amount of histone marks that you need to look at to find different classes of enhancers, for example, but we don't know that that's going to hold across other genomes. So there is value in a fishing expedition in this regard, I think. So the capped analysis of gene expression that I've been talking about, CAGE, relies on the fact that messenger RNA has this capped structure that can be used to pull out the messenger RNA, that you are then only sequencing the first 50 base pairs or so into the messenger RNA. So you're not able to look at cassette exon structure, but you are able to deconvolute internal start sites which would be hidden from short RNAs being contigued together. And this gives you a very quantitative and reproducible map of the proximal promoter architecture as well as a digital count of gene expression. And it's cheaper than RNA sequencing because you don't need to cover the entire transcriptome. The complexity of what you're looking at is much reduced. It's extremely quantitative. So here I'm showing you in the macrophage series that we ran, here is the pattern of CAGE expression on 3 donors, and here is the PCR validation on 6 donors, including the original 3, and you can see that the pattern is very faithfully maintained. I think the other reason to look at transcription is that we were able to start to show that there are very few housekeeping transcripts encoded in a mammalian genome, and I think this will be even more interesting from a comparative perspective in the animal kingdom. I'm going to skip over this aspect in the interest of time. Only just to say that when we had a look at alternate transcription initiation in macrophages, when we looked at the behaviour of monocytes responding to different pathogenic challenges, we were able to see fairly reliably, a core set of 200 odd genes which were always turned on. But there were many, many more transcription start sites being expressed in these cells, sometimes 3 to 4 times more. And this tells us that there's a lot of diversity being able to be driven from the genome, even with highly stereotyped signalling pathways such as the innate immune pathway. So in resting monocytes, we saw about half of the genes were expressed from a single start site and the other half from multiple start sites. But when we looked at activated macrophages, we found that the majority of inducible genes used multiple start sites. And without going into a lot of detail, what we were able to show is that in many cases, these drove differences in the open reading frames, particularly in the N-terminals of these open reading frames, which meant that the products of these genes were being located to different regions in the cell, secreted in a different pattern, or interacting with partner proteins differently. Some of these multiple transcripts drove convergent patterns that did not change the open reading frame, and I think that this is an important point, because what it was doing was amplifying the signal and giving the gene more opportunity to be expressed from many different regulatory regions. But in other cases, we saw very consistent pathogen-specific responses, and these often did drive different proteins. So on a pathway level, what you can see, this is the TNF pathway, that in yellow, you've got the genes with multiple transcription start sites driving what we predicted different protein products, and this has really got the potential of diversifying this inflammatory pathway dramatically. So again, coming back to this idea of expressivity of traits, from these genes we're driving diversity in the innate immune function, some of which is pathogen specific and some of which is not. So I think just to reiterate what I wanted to tell you about the value of your program to genome biologists like me is that there are some big outstanding questions in terms of RNA. And in particular, I think we need a new RNA ontology. I think we need to stop talking about coding and non-coding transcripts, because it makes the assumption that the only role of a coding transcript is to produce a protein, and clearly RNA is catalytic regardless of whether it can be shuffled to the ribosome. It also gets us around the fact that we're really crap at identifying open reading frames which are smaller than 30 amino acids, and John referred to the peptide coding potential of non-coding RNAs last night, but also many messenger RNAs encode upstream open reading frames in 5' UTRs, which may also be contributing to this peptide repertoire. So we need to be thinking about RNA ontology more in terms of its activity within a canalized network, its capacity to build the building blocks, its capacity to regulate and buffer genetic or environmental perturbation. So this comes back to this idea of integrating ideas on phenotype, expressivity and penetrance, removing ourselves from the strictly deterministic path, and thinking about how some of these non-coding RNAs might be buffering or increasing probability of a phenotype. And I think that there is strong value in looking at a transitioning genome in cells responding to something, as well as the static homeostatic tissue state. So the acknowledgements that I really wanted to highlight from FANTOM, the major people involved in FANTOM are listed here, in particular Piero Carninci, who's the big technology driver in FANTOM and the inventor of CAGE, Yoshihide Hayashizaki, who remains a very inspirational figure, Al Forrest, who led the most recent FANTOM 5 paper, my group, who've been involved in various aspects, and people at the University of Queensland who contributed to the monocyte study that I talked to you about. Thank you.
For a very interesting and broad covering talk. I'm sure that people have got questions for Christine. Surely someone wants to make some comment about bioinformaticians and biologists communication. The topic dear to my heart.
So Christine, very nice talk. Thank you very much. Maybe this is not your intent, but let me see if I can contrast with what John said last night. One thing that he said was, get the basic data out first. Healthy animals or healthy individuals, and don't worry about disease individuals. Don't worry about lots of environmental effects. Get that data out first, and then that's going to actually be very useful even for the other things. You would argue, maybe you're arguing, at least what I took away from it was, variation is important, collect the data on lots of different conditions and so forth, because it's all very useful, at least at the RNA level. Could you maybe comment on my perceived difference?
So I agree entirely with John's perspective that having a baseline map of normal means that you can assess potential regions, regulatory regions, and you can make predictions or hypotheses about what sequence variants might be doing, for example. But we must not lose sight of the fact that the genome is extremely contextual. And if you remove that context, then you're removing the majority of information that you're looking at. And I think in some ways, this idea that there's a reference comes from genome sequencing rather than really understanding genome regulation. So I would strongly urge you to set aside a set of tissues which are commonly of interest to the group and have that standardized set, but then allow your collaborators, your interactors, to bring to the table their particular biology, because in FANTOM it was those renegades who actually drove a lot of insight. And in particular, the macrophage libraries that we contributed to the first round of FANTOM were incredibly important. Full-length cDNA analysis relied on a subtractive method to try and enrich for new transcripts. And actually it flatlined until we started putting in cells which were responding to different cues. So homeostasis only gets you so far. It depends a lot on the questions the individuals who are contributing want to ask.
This is David Burt. I agree with that sentiment completely. Actually, most of the tissues last night weren't normal. They were cancers and oncogenic cell lines, so they're not normal. In fact, most cell lines that are established are not normal. Lucky enough, most of the tissues that we're going to work with, we don't have many cell lines anyway. So we've got to work with real cells and tissues. I agree with this idea that looking at all sorts of tissues. One of the problems with this community, unless somebody comes up with a huge amount of money to fund a central set of cells that we're all going to work on, the truth is that this is going to be more like a cottage kind of thing, where we're going to have not one or two major grants. We're going to have maybe 50 sort of grants. Each of these grants is going to have to be justified to their funders. And they will be justified, the sort of things you're talking about. There'll be one group, for example, that might say, I'll get this money to look at these immune cells because of, like Jerry was talking about, some sort of infection process. Someone else will be looking at development and things like that. So I think it's, we've got to cope with that. And I think the word flexibility someone's used is really what we're talking about here. So one of the challenges is how we're actually going to pull all these things together, because we're not going to have 20 standard cells.
So maybe the only other comment to add there is that it may be less important to do lots of stuff to all of those things in a centralized way. It may be more important to have a smaller set of assays that are going to give you a lot of information that's standardized. But if the funders are in the room, please, let me emphasize this again. This value is not just to the individual animal communities, that without this effort, we are hamstrung in the Human Genome Project at understanding these very difficult questions of network robustness and phenotypic penetrance, because we simply are not able to ask these questions in a meaningful way with the human resources that we have. And mouse isn't doing it. We have to be getting diversity of genomes to be understanding these fundamental questions, and it is going to have the same breadth of impact on understanding human traits that it will have on understanding individual animal traits. So there really needs to be some vision from the funders too here.
We agree. Any more questions? No.
Can I have one more?
Yeah, you can
Let me have one.
Yeah.
So, really loved your talk. Inspirational. You didn't talk about RNA editing.
No?
How important is RNA editing in terms of associations with phenotype and disease in human?
Look, clearly, both genome editing and so through retrotransposon activity, for example, and RNA editing will impact on specific phenotypes. But it's very hard, I think, to know how much of this is stochastic and how much of it is genuinely driving a heritable trait, because this is all happening in somatic cells. And it's hard to predict. A lot of the work that's coming out on RNA editing, I think, is biased by the technology that's been used to drive it. And so I think the jury is still out to some extent on the extent and importance of this in terms of RNA structure or function. Nevertheless, I think that a functional genome has to drive diversity even when the genotype is relatively static, and that these are clear mechanisms for driving that diversity. So for that reason, it must be important.
Just an extension on the sort of encouragement of funding agencies. I think many of the people here, if not everyone here representing the funding agencies, understand the issues. I think a bigger challenge for many of us in this room is actually educating the people that constitute the review panels. Because that is where many of us suffer, particularly when, I'm going to leave it at that. But I think we all understand what I'm saying, and part of the issue is we're simply not doing a good job of educating our peers.
So I don't know how we remove the drive to mediocrity in that situation.
Christine, what I'm wondering is how much value is there in deep phenotyping of the individuals in which you're doing? So all those off measurements that may not be directly applicable to the question in which you originally designed it.
Thank you so much for asking that question. I think it is extremely important because at the end of the day, what we're building are correlates between these molecular events and the phenotypes that we're interested in. And the deeper the phenotyping available, the better those correlates are able to be built. And if we're thinking about phenotype, it's very hard to assess upfront which phenotypes we should be looking at. We had a teleconference with FANTOM this morning where this was exactly the conversation and discussion around what phenotypes should we be including. So again, I think this is where having a strong biological community means that you're going to have much deeper insight, because the phenotypes important for chickens, for example, are likely to be quite different to the phenotypes important for cattle. And so as much as you can encourage that deep phenotyping from your individual communities and collate it in your databases, the better.
So just in defense of funding agencies, we cannot fund proposals that we don't see is one thing. So I've been railing at this group in particular about broadening your perspective and really engaging in different funding agencies. And emergence, robustness, and modularity, those are exquisite NSFE type questions. Milk yield probably is not. And so even if it's the same underlying approach, rationale, and study design, you have to learn how to use the appropriate language when you approach the different funding agencies. And bringing this group together, really, it's important for people to understand that each of the agencies, we do have a job we're supposed to do. And to Carl's point about educating the panel, look around, folks. The people who do it to you are in the room. And you can engineer a mutiny. If 10 or 12 proposals come in, we have to stock the panel with the right expertise to review those proposals. And so engaging at one level as PI is helpful. But then if you turn down the request to serve on panels or do ad hoc reviews, you're still leaving it up to somebody else. So this idea of getting the community to organize, engage, and coordinate is hugely, hugely important. It will not happen unless you come together on all this. That's just one panel manager's perspective.
Steve, I have to challenge that. We're not in the room. I haven't served on a USDA panel in 10 years. And the reason I haven't served on a USDA panel is I'm conflicted with everybody in this room. And because I'm conflicted with everybody in this room, I can't review your proposals and identify the best ones. And so there is actually a negative selection against the best scientists to serve on USDA NEPA panels. And I've told Sonny this, and there appears to be no way to change that internally.
So I'll just say we did one pilot study at NSF where they called it a mechanism design, where instead of avoiding conflict, it was maximized. Everybody who submitted a proposal had to review 7 other proposals in that competition. And you were judged, at least in part, on the quality of the reviews you provided and how those things came out. So we are experimenting with review processes to address exactly that point at NSF anyway.
Another troublemaker here. We actually see the same thing in the UK. Maybe I'm getting old now, but yes, that's right. For a long time, years and years ago, I volunteered to go on various committees and panels. And the ulterior motive was obvious, was A, to find out how it works. And it's amazing how many, particularly younger, getting ages in there, younger scientists think it's a waste of time. Right? It's wasting my time. I should be in the lab doing this X, Y, and Z, and not going on to a committee. But unless you actually do that, there's several things that don't happen. You don't find out how it works. And B, it's dominated by, I'll attack another community, all the plant, in the UK anyway, all the plant geneticists. And funny enough, the plant geneticists do really well, right? So there's a lesson there. So I definitely would say, do your time, do a couple of years of it or something like that, but encourage all your postdocs to do it because it's good for them. And it's not a waste of time. And by the way, I do, in the UK, I do review quite a few grants around here, and I assumed that many of you review some of ours as well. That we're all part of the same community. So I'm quite favorable to livestock genetics. So I think it comes the way.
Yeah, we've still got a couple of seats down here if you prefer to sit down rather than stand up. So are there any more questions going back to the scientific side of the presentation?
So the comment about the breadth of species to apply, one thing that they did in the sort of the vertebrate community where they were looking at transcription factor, motif conservation and so forth, and the work of Kristen Limbredt-Toe and others, they emphasized the breadth across the evolutionary tree. Is that something that should be a major consideration for us when we think about the species that we should be proposing to do on the functional annotation side?
Well, I guess if your motivation is to understand the evolution of those traits, then absolutely. But you also have a huge range in form and function. And if your aim is to understand the common rules that drive phenotypic expressivity, then I think that you have plenty of diversity.
Any additional questions? Please join me in thanking Christine once again. And so next up, we have Laura Clark from the EMBL EBI. She is also the co-chair of the Metadata and Sharing Committee for FANG.
Hi, so I'm Laura, and I'm here to talk to you about the work of the bioinformatics and the metadata committees within FANG and what we're sort of dealing with. And I suppose there are three main themes to this. One is communication, one is sharing data, and one is data standards. So from a communication perspective, communication, of course, is essential for FANG, and for FANG to function, we all have to be talking to each other. In the sort of data analysis and data sharing sort of side of things, we have two committees, the Bioinformatics and Analysis Committee, that of itself has 4 subcommittees to address the different sort of assay types and analysis challenges. And then we also have the Metadata and Data Sharing Committee. And these committees meet, sort of have teleconferences regularly, have email lists for communication. Everybody who is a member of FANG is welcome to join and participate. And we've also recently set up a sort of shared wiki, so people can post minutes of meetings, protocols they are using, sort of analysis pipelines, and all of these sorts of things. And this is essential for us to be able to work together and learn what's going on. Yes, my mic has dropped. Thank you. So let me figure out the mic for a minute. There we go. And yeah, we really need the collaboration of everybody in FANG. And obviously, the metadata and data sharing group is already working with the assays and samples groups to try and pin down the right things to define. So when it comes to sharing data, everybody who's a member of FANG has ticked a box to say that they are pro data sharing, pre-release data sharing. All of this is sort of, again, central to the way FANG works. We have to share data both with everybody in FANG and with the wider community. One of the important things to recognize, though, is not only just the data. A FASTQ file, a BAM file, a WIG file is useless if you don't know where it's from, how it was produced. So we also have to share descriptions of our data, metadata. So the sorts of ways we're pursuing this, Elizabeth already mentioned biosamples, and the biosamples database at EMBL-EBI we feel is the right way to share sample information. It's a much better structure for sample information than most of the assay archives. It supports sort of related to and derived from samples, so you can go from donor to tissue to cell type. And if you have a pedigree, you can have all of the animals in that pedigree registered and connected to each other. It also has native ontology support, so you can be specific about the samples that you have submitted, which is more challenging in most of the other assay archive metadata structures. That being said, we do also need to make sure that the metadata in the assay archives is also good, all the sort of experimental details, PDFs where there's a lot of information, but some concrete single values, so they're easy to sort of pull back out again without having to download all the PDFs and read them all. And the metadata and data sharing group is working hard to define both sample and assay sort of metadata standards. And then of course we have to also share our standards. So it's all very well and good saying we've got standards if people don't know what they are. And we're following, I think we sort of borrowed this idea from the Global Alliance for Genomics and Health, and you're using GitHub, which is a sort of version control system and flat file text files that anybody can edit, and we can all sort of version and make sure that everything is moving forward in a productive but easy to find and easy to use manner. And of course, we must share data. Everybody, as I say, everybody signed up, ticked the box to say that they're willing to follow Toronto pre-release principles. It's great to see that we've started generating data. I will be really excited when that data gets into the archives. and we can tell everybody in FANG this data exists, this is where you need to go to find it. Of course, that's great for raw data, but analysis results, at least in the first instance, we won't want to submit to the archives because we don't know precisely how to do the analyses. So we do also need our own methods of data sharing. We have a public FTP site, and hopefully we'll be able to move to a situation where we can make regular releases of analysis results, of interpretation of this data that the entire community can use and can access and can visualize. We also have an internal data sharing mechanism because, again, we don't know what the right way of doing things, so we might want to run things 5 different ways in the first instance and then let everybody pick it apart and go, actually, was that right, or should we have done it this way, or would it be better if? And so the internal data sharing is very important to support that and to make sure everybody has access. Everybody within the consortium has access to data as quickly as possible. Of course, the scale of data we're talking about here has the potential to be quite big. So these things are all accessible via FTP that most people know how to use, but we've also enabled a couple of technologies that are good for big data, for fast data downloads on large data volumes, and that's Aspera and Globus Grid FTP. They have sort of slightly different interfaces. But that should mean that everybody can get access to even the very large files that end up being posted. So now the majority of the rest of my talk now is about standards, and we have to have standards. This has come out in both of the previous talks. If we don't have standards and people don't stick to those standards, then we're not actually going to be able to draw together all of the data we generate and produce meaningful interpretation and actually deliver biological insights. So the first set of standards are metadata standards, and this is being driven by the metadata and data sharing group. As I already said, we're using GitHub to track these things. We have text files that are just defining the principles. We're also going to work to set up validation services. So not only can people see what the standards are, they can check if their own data has met those standards. The way the sort of metadata is sort of being viewed is there are 3 different types of metadata. There's sample metadata, so what the biological material is. There's experimental metadata, what the RNA-seq, ChIP-seq, whatever other experiment is. And then analysis metadata, how did you take that sequence data, map it to the genome, process the signal, turn it into more useful information. One of the important things here from a sort of data description point of view is that we use ontologies. This is particularly essential for samples, but is also important for experiments and analyses of objects. And this is so everybody can have a common understanding of what we mean. When you say macrophage, or you say monocyte, or you say CD4 T cell, these can have variable meanings depending on how it was sorted, what the cell surface markers you're using, all of these sorts of things. So by having specific ontology terms to describe what we've done, people can understand that you are talking about the same thing. And luckily at the EBI, we work very closely with Helen Parkinson, who has a very strong understanding of ontologies and drives a lot of ontologies like the experimental factor ontology. So we're well placed to support the community doing that. But of course, there are lots of ontologies that within the EBI we haven't already dealt with, like the animal trait ontology. And so we need everybody to come and say if there are terms that don't exist, how to define them, or there's a type of feature that we're not using that we're not covered by an ontology, what the right ontology to use is, because we need biological domain expertise, and that isn't always present in the sort of bioinformatics side of things. And one of the advantages of using Git to share these things, obviously, if you want to be a member of our Git organization and you're comfortable with doing it, that's great. You can access it sort of on, traditionally Git is thought of as a code repository and everybody uses it on the command line. You can totally interact with Git through the website. You do not have to download any clients. You do not have to know how to edit a file on the command line. You can hit a button, a plus button or an edit button in the web interface and start changing the files. And you don't have to worry about getting it wrong because all of the history is tracked. It's very easy to use. And everybody's welcome to contribute. If you are a member of our organization, it could be done in a more easy way. But even if you're not, you can still propose something. Someone else will have to validate it and accept it, but you can still interact. And I think this is vital to make sure that there is totally open communication about what our standards are, and everybody can suggest improvements to those standards. So then we go to data standards. I mean, this is somewhere where there will need to be a lot of discussion. There already has been sort of issues about this raised already today. Possible metrics, and please note, this is a starting suggestion list, not the definitive list of metrics we should use. Of course, you've got uniquely mapped reads. I think I meant to add PCR duplicates to this slide, but obviously the set I sent in, I'd forgotten to add that. And then there are metrics that are appropriate for specific assays. So strand cross-correlation for ChIP-seq, where you're making sure that each strand has about the same number of reads mapped to it. Bases mapped to coding regions in RNA-seq, and of course, RNA-seq metrics will have to take account of the completeness of the annotation, but at the same time, not all mapping reads that don't map because they're missing genes. Some reads don't map because they're poor quality data. So we have to bear in mind these aren't straightforward binary values. And you need multiple values and to set the standards appropriately, so you don't throw out good data after bad. And then for sort of methylation, you'll things like base composition and conversion rates, making sure that the actual bisulfite treatment has worked and you're getting a good assessment of the methylation state of the genome. This is a sort of standard reference data sets is something that I'm sure anybody who's heard me talk on the analysis calls will know I hold sort of very close to my heart. We need to make sure we have standard references. If people are using different genomes, even different versions of the same genome, we get different answers, and you don't have any interoperability, which basically spells the sort of doom for actually integrating all of this data. So we need standard reference genomes. Ideally, they will be the genomes that the browsers support, because that means we can visualize the data without having to build new tools to visualize the data. We need standard gene sets. Obviously, I work very closely with Ensemble, and where there are Ensemble gene sets, I think we should use the Ensemble gene sets. Again, and then if we have blacklists or other sort of regions of the genome that we are more suspicious of, or ways of filtering the data, we need to make sure those are also shared and also standardized, so everybody's doing this in the same way. Of course, there will be new assemblies. I think all of the genomes that FANG are currently considering having new assemblies built for them, possibly multiple new assemblies built for them. So we will need to make sure we have a plan for how to move, which genome to move to, and sort of to make sure that the capacity is available so all of the data can be remapped and reanalyzed. Because you can't have the old data on one assembly and the new data on a new assembly. Again, it sort of doesn't work together that way. But at the same time, when we do move, say you define a pipeline, we define a pipeline in the first quarter of next year, and then the first quarter of 2017, we have new genomes for everything. There'll probably be new methods. There'll probably be better versions of existing methods. We can also take that as an opportunity to actually reconsider our pipelines and go, maybe we used to use, STAR, maybe now we should use the newest, shinest RNA-seq aligner instead. And so it's not just a challenge, it's also an opportunity to improve the way we do things. But again, this comes back to the communication. We all need to be talking about this, and we need to come up with a plan and stick to it, rather than, obviously there will be cases where people have specific questions that will want specific ways of doing things that, for a particular paper. But for the centralized things, the things that everybody uses, they need to be done in a relatively standard way. And this is where I think probably one of the main topics for the data analysis discussion this afternoon will be how we standardize pipelines. Is there a single pipeline that will actually work for every assay? The answer to that is probably not. There will be multiple pipelines that will all produce answers that are sort of vaguely comparable. Actually, which one of those is the best is a very difficult question to answer. So you have to, on one level, be pragmatic. If there are 2 or 3 centers doing the vast amount of the analysis, and those 2 or 3 centers all agree on one pipeline, but someone else has another one, maybe the standard pipeline has to be one that the centers doing the majority of the analysis use. But we do also have to recognize there will be times when a specific pipeline for a specific question is the right solution, and we shouldn't stop people doing that. We should enable that, and we should make sure our ways of describing pipelines means that the unique pipelines can be as easily understood and as easily described as the standard pipelines can be. One thing that we should probably investigate, there's a new sort of burdening burgeoning standard called the Common Workflow Language, which is a way of describing pipelines. And we should look at that and other similar solutions to make sure that we can actually give people easy to understand information about the pipelines. So I think that is my last slide. So please let me know if you've got any questions. And I just need to get some water.
Laura, are you going to be mapping to viral genomes as well?
So, I think some kind of contamination screening would be a good idea. At the same time, mapping to the reference genomes, those viral contaminants and other things tend not to be there. So don't map. But yeah, certainly considering sensible contamination screening is a good idea and something we should think about. Obviously, a lot of places run things like FastQC, which do a certain amount of that sort of out of the box. But yeah, we should think about it.
And if there are lots of questions, please feel free to take advantage of the microphones on both sides as well.
I can just talk loudly, I think.
Where's it?
Okay. Hi, Laura. How you doing? Just to reiterate, I think it's a great presentation. Just to say, I completely back you up on this. I think adoption and use of standards is probably the most important part of this project, because if we don't adopt standards and use them, when we compare results, not only will we find biology, but we'll also find a lack of standards in our results, and we won't know that's why we found those. So we absolutely have to do this. It's going to be really, really difficult.
Okay, we have time for maybe one more question or comment.
Thank you. So this issue of contamination is really difficult to deal with, because all of these projects are actually metagenomic projects, right? When you harvest RNA from the tissue of an animal and you sequence the RNA from the tissue of the animal, you're sequencing everything that is in that tissue, not only the animal's RNA. And so we've just done this for the reference cow, and both the unmapped genomic reads, we have 70x coverage of Illumina paired ends, and of course about 7% don't map. Same for the RNA-seq data, 17 tissues. You get a whole bunch that doesn't map. A lot of that is the stuff that's missing from the reference assembly. Then you start finding pathogens. You start finding nematodes. And there's a bunch of nematodes in there, and one of them turned out to be Onchocerca achengai, which actually lives in Africa and doesn't live in the US where this animal came from. And the reason that that pathogen turns up in the sequence is that the actual reference sequence for Onchocerca achengai is contaminated with cow DNA. So you end up with this enormous complexity of finding things that you say are contaminants. And yes, they're contaminants, but only from the perspective of they don't originate from the actual cells of the animal. They may very well be living along with the animal that you've sequenced.
So probably a good parallel to that, for the 1,000 Genomes project, we have, it's all LCL, so lymphoblastoid transformed cell lines, which are transformed with EBV. A decision was made early in the project to include EBV in the reference that was mapped to, because there are people who are interested in, humans have native EBV sort of things in them, and by mapping to a reference that included EBV, we're enabling the biologists who are interested in that angle of the genome to also see that. But it also acts as a decoy, as a sink, to pull out all of the reads that are sort of there from the lymphoblastoid transformed cell lines. So if for particular species we are aware that there are very, very common sort of metagenomic sort of passengers, we could consider adding additional sequences to the references for reference FASTA files that we distribute to help with that. We should also do contamination analysis just to find out, because yeah, I mean, best will in the world, if you don't quite do your pipetting right, sometimes you can end up with things you're not expecting in there. And yeah, we have to be cognizant of that and make sure that all of our processes deal with that. But I think by discussing the issues and defining the correct standards, we can manage this relatively easily. So,
thank you very much. Please join me in thanking Laura once again.
I'd like to introduce our next plenary speaker, who's just made it from Baltimore. So please welcome Paul Flechet, head of Ensemble, involved in many large genomics projects, and also running a research group looking at the evolution of regulatory elements in mammals.
Okay, thanks very much. I'm actually really happy to be here today, and I also recognize that I don't know too many people in the room, just maybe a few of you, and so I was really happy actually to listen to Christine's talk, partly because it hit on many of the points that I want to hit on, and it came from a different perspective, and so I hope by giving you my perspective on some of these same things, we'll see that actually there's a lot of common ground here. For those of you who don't know me, I am a bioinformatician, a computational biologist from the genome at the very beginning. So I discovered computational biology when the human genome was being sequenced, and a computational biologist named Sean Eddy was giving a talk, and I thought, wow, that's way more exciting than anything else that I have the opportunity to do. And so that's what I've been doing for the last 15 years. What I want to talk about today, actually, and this is what Chris asked me to talk about, is rather than just sit here and talk to you about what we do and what my experiences are, to try to get discussion going about ways to streamline things and get analysis going. Now, as a computational biologist, I've given many talks to audiences that end with no questions whatsoever. I think this is probably one of the most common things for a bioinformatician, to give a talk with no questions. I assume that it's because either people don't care about bioinformatics, which may be true, they aren't really all that interested, or they have convinced themselves that I am not insane and they are happy to let me do the computational work and kind of get about my business. So I don't want 30 minutes of silence, although maybe some of you want an early coffee break and then it would be okay. And so because of that, I'm going to do a couple of things. I'm going to consider infrastructure beyond hardware and software to include social infrastructure. Christine touched on this. It's incredibly critical. I'm also going to be opinionated, and I hope this sparks some discussion. If it does not, well, that's the way it goes. But I would say, and Laura hit on this as well, and Christine hit on this too, talking early and openly and an honest commitment to working for the common good is critical. It's the only way this project will work, if we are all committed to the common good, to a common goal. So some of the concepts, and I'm going to try to give you some of my worldview here, and some of the concepts that I think are important. And I should say these come from, well, I'll tell a couple of stories as we go along, but it comes from trying to understand and manage data sets and data in large-scale projects. The 1,000 Genomes Project, Laura, who was just up here, was critically important in that. And it also comes from trying to run community resources like Ensemble. Lots of people use Ensemble. Lots of people have opinions about Ensemble. We try to make it as good as we can. And the process of doing that informs a lot of my opinions. So one of this is to make things as good as possible, but don't wait until they're perfect. Understand and agree on the overall goal. Where are we going? What does it mean to get there? And there will always be new things, always coming. PacBio sequencing will make the assembly better, absolutely. Oxford Nanopore will enable us to do something cool, for sure. There will be new ways to do experiments, to prep things, to do everything. If we wait for all of those to happen, we don't discover anything. And so to get started. 1,000 Genomes Project just finished a week ago. It took 8 years. The initial planning said it would take 3 years. Hey, but it got done. And actually, along that process was a lot of good things along the way. But it's important not to wait till it's perfect. Rapid pre-publication data release occupies the scientific high ground. This is something that a lot of people don't want to talk about. They want to keep their data. They don't want to release their data. In our current world, where sequencing genomes can take place anywhere and at capacities that are completely and utterly unheard of, data richness doesn't matter. Everyone is data rich. Analysis is where all of our bottlenecks are. And so I'm just going to be honest here. If you are sitting there thinking we release these data, there are 1,000 analysts waiting to jump on it and scoop you, you were wrong. Because those analysts have data piles so big behind them that they will never finish analyzing their own data, much less come and take yours. Now, can you come up with some scenario somewhere that something happened that made people uncomfortable? Absolutely. But I would say that in almost every case that this has happened, maybe in every case, this is a sign of incredible success. The 1,000 Genomes Project was so successful that people did some analysis that made a few people in the 1,000 Genomes Project uncomfortable. Well, who cares? The 1,000 Genomes Project has been the most successful next generation sequencing project. It created the standard file formats, it created the standard analysis tools, and it's defined how we do many things in next generation sequencing. So there was a preprint posted to bioRxiv that used some data a little bit before it was published. I don't think that was a big deal. Learn from the large scale and collaborative projects around you. So know what's special about you. I'm going to tell a real story about a large scale consortium that I was at one of their early meetings. Okay, I'm just going to say it. It's the International Cancer Genome Consortium. And they sat around in a room like this to decide what the best alignment and variant calling tools were to use. And I raised my hand and I said, the 1,000 Genomes Project has just spent 6 months doing this. The person at the front of the room said, that's not valid. Cancer DNA is different. Because cancer DNA has J's. See, this is what I realized. It has J's. There's 5 letters in cancer DNA. No, it's not. See, they spent 6 months doing this because they felt like they had to. We don't have to do this. We can learn from the projects around us. And engage, buy in, and accept that FANG is a long-term relationship. Marriage is a useful analogy here. And to get this process done requires time, effort, and understanding. So these are some of my biases. When the ENCODE project, the ENCODE main project, ENCODE 2, I think, is ENCODE 2 a good way to put it? Was published, Ewan had a commentary, Ewan Burney, who now the director of the EBI, led the analysis group. He had a commentary on lessons for big data projects. And there's a number of things that he wrote about here. The working for the common good is probably the most important one. And I would encourage every one of you to read this commentary and reread it regularly, but also understand that it's not the only way. The 1,000 Genomes Projects did some things differently, some things better, some things worse. This is true of all big projects, but a lot of the lessons and a lot of the concepts that Ewan talks about here are critical. ENCODE had to resort to a lot of written rules. The 1,000 Genomes Project had basically no written rules. But the commitment to the common good is the key thing that sort of drives through this. Okay, so I said I do informatics. Informatics is not one thing. Computational people are not one set of people. There's I feel still much misunderstanding about this. I'll get the computational person to do this. It's like saying get the scientist to do it. There's a huge variety of what we do. Everything from discovery-based biology to understanding and doing things, what some people say, blue-collar bioinformatics, down in the plumbing. So I've kind of thought about this blue-collar bioinformatics. I understand the sentiment there, but there is aspects to the plumbing analogy that I just don't like. Fixing leaks, carrying waste away, all those sorts of things. I would like to propose a different analogy, and one that is closer to engineering and building things. Because, and this is actually a picture of the construction of and the completed version of the Millau Bridge in France, which is one of the most spectacular things that I think has ever been created. It's the tallest bridge in the world. It's one of the longest. Elizabeth knows all about this. Okay. But some people sort of think, well, only the French are going to build something this big and grand. For those of you here, of course, it's important to remember that Washington, D.C. was designed by a Frenchman, which is one of the reasons why it's such a big and grand city. But this bridge was designed by Sir Norman Foster, who's British. See, you can have collaborations to make things work. And building things is not necessarily, and making things work is not about plumbing. You can build and make things work that are among the most amazing things in the world. And solving engineering problems can create incredible things. And it's important not to just say, right, we're going to go one incremental step. We need to look to the future. And this, I think, is very exciting. And I find these sorts of challenges and problems exciting. And so, yes, we have to do some incremental things, but we should look farther and do some bigger things. Okay, so let's start talking a little bit about, not put up $135,000 electric sports cars and start talking about the real question at hand here. 1,000 Genomes Project is an area where we at the EBI, me personally, Laura Clark, other people learned a lot about data coordination and management and the importance of orchestrating, the importance of having a role that orchestrates how things work and takes things from raw data to public release and supports the project in getting things done. And so I just wanted to put this up here. I'm going to describe some of the things that we did in the context of the 1,000 Genomes Project, and then come back and talk really about some of the social aspects of the DCC as we go a little bit further along. 1,000 Genomes Project started the original plan. The original plan was described in 2007. We got the first data in 2008. When the first data started to arrive, we hadn't hired Laura yet, so it was me doing it, which is one of the reasons why it went so poorly, I think. The data was coming in, it was breaking everything we had about the EBI. We couldn't copy the files successfully from one file system to another. We couldn't get them loaded. The data was being generated at the Sanger Institute, which if you have been to Hinkston is next door to the EBI. We couldn't transfer the files between the two. We eventually ran a single network cable from one data room to the other, and we punched a hole in the Sanger firewall. And I had the head of Sanger Systems come over and point his finger in my office saying, Paul, don't F this up. Because we just punched a hole in our firewall. So this is what it felt. It felt complete catastrophe. Today, of course, things run smoothly. The type of data that took us 2 months to process in April 2008, we do every single day now, without even thinking about it and without worrying. And we learned little bits of internet and other infrastructure that's important to know, but it's important only to know so that we can hide it from everyone else. This is an example, actually, of our first major data transfer. We had bought the software Aspera. Laura referred to this in her talk, and we were sending data to the NCBI. This started on a Saturday afternoon, and I pushed the first button. And you can see this is all the data moving out of the Hinkston router. And this is the long-term average at that time, was not very high. And it jumped up and went on this plateau, and it ran all weekend. And I would log in and look at it. I thought it was the coolest thing in the world. That where it drops on the front is 9 a.m. Monday morning. I'm convinced someone walked into the network operations center somewhere and said, oh my God, and they shut it all down. It took us about 8 months to get it running this smoothly again. Not quite, but it took a long time then to correct it again to get this working. Now, of course, all of these things run really smoothly, or much more smoothly than it did at the start. And so this comes to kind of the major message here, that infrastructures are critical. They're critical for everything we do. Ewan Burney likes to say, none of you called the National Academies this morning and says, hey, are the lights on today? Do you guys have electricity? You don't even think about that. You just come because it's going to work. And the only time you ever notice infrastructures is when they fail and when they break. And so we have this sort of amazingly difficult challenge when we build informatics infrastructures, is to be successful, is to be completely hidden. Try to go to a funding agency and say, we need money so no one notices we do anything at all. It's a challenge. But it's a challenge that actually, over time and with the right arguments, we can actually win this sort of argument. So I'm going to talk about informatics is infrastructure in a couple of different ways. So this is the single chart that defines the entirety of the European Bioinformatics Institute. It's our data growth chart. Over time, and through some process that is essentially indistinguishable from magic, the cost of hard disk storage has fallen about every 18 months. So the amount of hard disk storage you can get for the same price doubles every 18 months. This has been going on much, much beyond anything anybody thought was possible, and it continues to go on. And so this line, this 18-month doubling time, is our ideal line. It means we can spend the same amount of money for all time and store all the data that gets generated, as long as we stay on an 18-month doubling time. 12-month doubling time, more challenging. We can maybe work with that. 3 months doubling time, the entire building explodes. And so to move things to this 18-month line, or as close as possible, is one of the reasons that we exist at DBI, or one of our major infrastructure challenges. We have a number of ways we can do this. And so we can do this with things like data compression. There's a lot of ways you can compress data. Sequence data itself is very amenable to data compression because there's so much commonality between individuals of the same species. And you can do this philosophically in a couple of different ways, horizontal or vertical compression. So here's an example of, this is the type of compression that's used in JPEG. And again, here's a case where in informatics, we're borrowing from another field. We didn't invent this. We had to tweak it for sequence data, of course. But these concepts exist because they're needed in other places. Horizontal compression here just bleeds out resolution all the way through. That's basically how JPEG works. Vertical compression, I hope you can see this, blurs out the background while keeping the interesting parts at higher resolution. This is how video compression works. If you're watching a streaming baseball game, they're not refreshing the green of center field constantly as much as they are the batter's arm, for example. And there's nice algorithms that take place in all these sorts of things. For sequence data, vertical compression is perfect because the stuff that's the same across all the individuals, you can compress in a different way than the stuff that's different. And that's a logical way to approach this. Standards. Everybody talks about standards. Here's my slide to illustrate this. People like to give examples on some complex bit of phenotype that you need standards to describe. Here's an example with the simplest bit of phenotype we can find. This is looking through the European Nucleotide Archive database to figure out how many ways people have said female when they have put something into the database. It's about 90 different ways here. This is fine if you're reading all of them, because you can tell that this is a misspelling or something like that. Your computer cannot do this. Your computer gets very upset at this. And so to get these standards right is critical. Okay, I'm going to talk a little bit about moving towards some of the social aspects of data coordination. And I would describe the social aspects as everywhere you have one of these yellow arrows of data flow and starting to move things beyond. There's other parts of informatics, of course, that I alluded to earlier. Here's algorithms and integrative analysis, where we start to get the biological concepts out of what we're doing. But all of these yellow arrows are where we move data around, and we make connections between things. And so this comes to what I would describe as the 4 stages of project data coordination. So stage 1 is usually at the beginning. This isn't really all that hard. It's a few undergraduates, some hard drives from Amazon. And then when you start to get into it, invariably, there's questions about, wow, that costs a lot. How can we get it to cost less? Oh, if it costs less, you can only do this little much. And the danger here is unrealistic expectations, which is, again, what people have discussed, what Christine discussed as well. Stage 2, this is where the data coordination becomes blamed for everything that is going wrong in the project. I am incredibly familiar with this. Laura is looking at me familiar. And this is where things go wrong in these projects. Things go wrong because it takes time to generate data. It gets generated incorrectly. The QC metrics that everyone thinks they've agreed on actually shows that Institute A produces high quality data, Institute B, not so much. And then they go back and look at their internal processes. These things go back and forth, and they take time to work out. And many people think it should be done in a slightly different way. This stage normally actually includes an intervention. where you are sat down in front of a room and told how to do things better. Stage 4 or stage 3, it does work, and we usually get to the point where it works. And this is where the best science gets done. Things are flowing, things are working very easily. We are enabling things. The road is built. The electrical grid has enough capacity to deliver what needs to be done. And here there's support from multiple constituencies. People generating the data, the people doing the analysis, members of the community, everybody is getting something of what they want. And then finally, stage 4 is the rewriting history, where it's suddenly so easy. But unfortunately, this stage is also characterized by unrealistic new requirements. And technology changes, especially in a project that goes on for a while. And these unrealistic new requirements can be a part of that technology change. So data size and complexity goes up, project duration extends, all of these sorts of things are happen once the data infrastructure has managed to make things, the challenges disappear again. This has also been kind of a huge theme already. Getting through the stages is about communication. So understanding how things work together and how we can all work together. There is of course, new people are coming into genomics and computational biology all of the time, and they're coming from other fields. For some reason that I don't understand, this causes consternation from some people. If you are involved in a field that is growing and is so exciting that it is drawing in people from other fields, as we are in genomics, we should consider ourselves incredibly lucky. We are blowing up groupthink. We are bringing in new smart people. We are like the Silicon Valley of science. We are collecting some of the most involved people. And so we should embrace this. But we have to talk to them. My goodness, if they've spent their times in the computer science lab, the way we do things in genomics doesn't always make sense. Some of it doesn't make sense even if you haven't spent your time in a computer science lab. But we need to talk to them. And so picking up the phone and talking to people, coming and meeting together, yes. Email, when you send an email, you know exactly what the tone of that email was. The mutations required in the FOXP2 gene, or whatever gene will be required for other people to understand this, haven't happened and spread through the population. So email is a notoriously difficult mechanism. I also have really strong biases about open data. And this goes into the EBI's biases about open data as well. Freely available data and resources drive innovation. Sometimes it's challenging to make that argument. The temptation to put a subscription fee or something else on a data resource is too high. These things trouble me. And so fortunately, the EBI were funded to do this. Other people are trying to make this point as well. I have no idea where the numbers on this slide come from. In fact, all of the numbers, I don't know where they come from. But it's a nice big number at the end. So I didn't, this seems like a high, I mean, if the government did pay that much for the genome, okay. But still, I think the general point there is fine. Finally, some things that we often hear. It should work just like Google or Apple. I fully agree. And if you give me the budget of Google or Apple, I'll do my best. But but let's be honest here, lots of companies do have the budget of Google or Apple, and they don't succeed at it. So aspiration, we have to have really high aspirations. We also have to be relatively realistic. I want a button that does all of my analysis automatically. I want a button that gives me more money when I push it. And so the goal here is to really communicate and come up with the best way to do things. And so finally, to kind of go back to this sort of thing about ambition, we can do things incrementally, but we don't have to. And big problems often need big solutions. Now imagine if you came into work one day and in fact somebody said your job was to write a cruise ship that has just crashed off the Italian coast in a protected area. This is not a hypothetical example. Right? It's a real example. And so it's a big ship. It's a big problem. It's hard. But you can do hard things. And solutions are often possible to hard things. Solutions are rarely free. When we can make solutions of low cost, when we can use things that we've done before. I mean, sure, if one of these crashed again, it would be cheaper, right? And they do it better. The second time is going to be cheaper. It's going to be faster too. Some of these things we do for the first time. Some of the things we do for the second time. As we can get the balance right and learn from the projects around us, where we can do it the second time, we will all benefit. Where we know in FANG we have to do it the first time, we should also specify that and make sure it's appropriately resourced. So this, kind of to wrap up, infrastructure enables discovery. You know, that it's necessary, going back to some of the other comments about review panels, can be conceptually unexciting, but it's absolutely critical to do the data management right. And to cross-justify the infrastructure with what will come from the infrastructure. And actually, there is a similarity to these Victorian sewers and, in fact, the Large Hadron Collider. You build things underground, send things around. It works. So really to conclude, I think about informatics and infrastructure as building these tools and resources to enable genomic science, but they do more than that. They create stability and they enable reproducibility, which is what we want. We also have to, and Christine had, I thought, an excellent really defense of large-scale genomics projects. The idea that you can do science one way in biology is preposterous. Science should be done in multiple ways and across the spectrum of projects that are larger and projects that are smaller. Larger projects create skeletons upon which we can do additional things, specific investigations or detailed things. And to make these connections and to do this in an intelligent way benefits all of us. But we need to understand each other. We need to communicate to create the real insights. So I have a couple of discussion questions because I remain terrified that I'm going to finish and people are just going to be quiet for a half hour. People in FANG have signed up to the 2009 Toronto Agreement. How many people have read the 2009 Toronto Agreement? Do you know what you've signed up to? Does that concern you? Right? This is about data release. In fact, it should be noted that you and Bernie, who is one of the key authors of the Toronto Statement, actually questioned, and he didn't explicitly refer to it as Toronto, but he did in his that 2012 commentary I put up, the clumsy etiquette-based restrictions. And etiquette-based restrictions are part of the Toronto Statement. We need to think about how we deal with this. There are some things that scientific etiquette remains important, but as we continue to move further and further into this massive data-rich, analysis-poor world, we can take the high ground in different ways than we did before. So the second question is about what makes us special. What new analysis tools do we need to develop? Do we need a specific short-read alignment program for cattle genomes? Is cattle DNA different? I hope not. And then what are the most important questions? This also sort of calls back to how do we set up the infrastructure to deal with the most important questions? You can build a spectacular highway, but if it goes where no one wants to go, it doesn't help anyone. And to get these discussions going early is key. Okay, that's really all that I wanted to say. I hope this was a useful discussion. I hope it stimulates some additional discussion going forward. So thank you very much.
Thanks very much, Paul. Who wants to start the conversation?
I have a question. So you talk a lot about the infrastructure that has been built maybe for other projects. So is there a need to build more infrastructure, or is it enough with just expanding the existing one?
I think partly this is a question of really understanding what the overall goals is and where things are going. You know, we can draw comparisons, and comparisons are really valuable because they help us learn. But FANG is different than ENCODE or the 1,000 Genomes Project. And I'll give you a couple of reasons why it's different. ENCODE had a coordinated funding structure that gave out grants, that had, Elise is in the room sitting right behind you, had strong program leadership involved in the project. That has been critical to ENCODE's success. The 1,000 Genomes Project almost had no grants associated with it. This was just paid for mostly by people excited by the project. And then taking from the major center grants that powered the large-scale sequencing centers in the US and other places. But the 1,000 Genomes Project had a different type of collaborative history. This was essentially the same groups that had sequenced the human genome years before. And so they had established some of the relationships in the marriage analogy. I think FANG is different than that. FANG is building new relationships. FANG is going to be funded out of a process of different grants. This requires a justification to those funding agencies and a return, a traditional return on some of these grants in the form of publications and other things that are subtly different and sometimes importantly different. And so mapping out these differences will help us allow where we can reuse stuff and where we do things differently.
Does this work?
Yep.
Okay. Hi, Paul. Yeah, we may not need a different sequence aligner for chicken or cattle, but one can see that the single reference genome, single haploid genome representation paradigm is going to run its course relatively shortly. How soon do you see the representation of a genome for a species that in fact is a composite of multiple assemblies? How soon do you see that coming along? And where are we positioned for the tools to interrogate those assemblies?
So the answers to these two questions are interlinked. If you can create a representation of the genome that has no tools associated with it, it's a curiosity. Yep. We have no tools to align DNA sequence reads to microscope images of the karyotype, for example. Those tools will not be developed. It's too big a thing there. But the first versions of the graph genome, which apparently are going to be released next Tuesday at the New York Genome Center from the Global Alliance for Genomics and Health meeting, are the current equivalent, essentially, of an image of a karyotype on a microscope slide. There's no tooling around them. It is interesting to see this. It is a critical step forward. People are working on tooling, but until you can align something that aligns appropriately into a graph structure and in a way that gives both enough performance to do the calculation, it would be a computer science curiosity if we could do the calculation in 42 terabytes of RAM, for example, but not a practical solution. And we also have to test whether or not it makes a big difference. And this is something that it can be more correct and have very little impact on the underlying biology downstream. And so, other examples where people can argue, but it has relatively little impact on the biological insights is, what do you do with multiply mapping reads in a ChIP-seq experiment? Do you throw them out? Do you keep them? Does it matter to the end result? A little bit. But does it matter to what you're really saying? Probably not. So the graph genome is coming. The tools are a ways off. Putting them in effective pipelines. If we have that in 3 years, I will be really surprised. If we have it in 5, I think that'll be about right.
And what's the impact on the sort of best in genome paradigm for annotation?
So I think we have to keep that. This is what the tools have to figure out how to do this. If we don't sort of manage this correctly, let's say we have two individuals' genomes that we are analyzing together, and a best in genome process, because some RNA-seq data maps better to one haplotype than another, that doesn't mean that the gene only exists on the haplotype it maps to because it maps there better. Right? We have to have a process that understands best in genome, casts a shadow down to other parts of the graph that are related to it. And this is fundamentally a hard problem, to do this in a way that will be statistically robust. But we have to take a step back and remember, alignment programs are heuristics. They are not. It is still an approximation of the truth. Now it's one that's well controlled, and we understand the errors in there. But alignment programs are still heuristics. So this is where we're going. It's a hugely exciting area, and one that is fortunately for us in this room being driven by problems in genomic medicine and especially things like cancer sequencing. And so we should be able to piggyback on this in great ways.
We've said earlier, and I just wanted to reiterate, a difference between this project and the human projects that have all these other resources like the mapping tools that have defined protocols, is that the genomes involved here are draft genomes, and so there will be differences. So maybe you'll use the same mapping tool, but you'll definitely need different parameters and different evaluations of the results because of that.
Oh, that's exactly right.
So yeah, I just want to speak to you. We do. I think everybody in this room knows that these genomes are not of as high quality as human or mouse. I think there was a small livestock genomics meeting in Cambridge last year, and Dave Burt used the term drafty, which I think is quite apt. And yeah, we need to make sure all of our quality metrics and our how we parameterize, how we decide what isn't good and not, and which regions of the genome we can make reasonable biological assertions in. There may be regions of the genome, I mean, there are regions of the human genome you have to be a little tenuous about making biological assertions in because they're a bit ropey. It's very difficult to genotype people in the HLA, for example, just because the HLA is complex. And so, yeah, we have to be cognizant of that. But I think as long as we're open in communication and people, one of the wonderful things about the 1,000 Genomes Project was that anybody, even a sort of junior postdoc, could turn up and say, I think we're doing this wrong. This is the evidence. I think we should do it this way. And everybody, Richard Durbin downwards, would pay attention, look, would pick up out their arguments. And if they were right, we would change how we did things. And that I think was one of the amazing aspects of the 1,000 Genomes Project. And we have to try and take a similar, everybody should be able to say, maybe that's not right. So we don't put out information that ends up being wrong.
Paul?
I wanted, going back to the question of the single reference genome, I wanted to say that, that there is the model that the Genome Reference Consortium developed for the human genome, where you have a primary assembly and then a number of alternate loci for regions where there is significant haplotypic variation, and that is an intermediate between the single reference genome and the graph-based genome. For which there are some tools available. There is an AlterWare aligner now that can deal with that, where you actually have alignments between the alternate haplotypes and the primary assembly that can be used to establish the relationship between things that map on there. So, there is that model that would be available for livestock genomes if people want to go in that direction. Yep. Thanks, Paul. Very nice talk and got people thinking. Just a couple of comments. I'm part of, and you are as well, a group of people that are interested in multiple genomes. which kind of hits on some of the things we're talking about here. I mean, there's alternative ways of looking at multiple alignments. It could be between different species, which we're quite used to. Think of a reference genome that we compare to that, like human or chicken or pig or whatever. And conceptually, it's very similar in a way to looking at multiple genomes in the population. So I don't know how things are going to move, but Benedict Patton, University of California, Santa Cruz, has got a system called Cactus, which allows you to, the focus at this point is to look at multiple species, multiple genomes. And of course, it all depends how you compare them. And again, it's a bit like your reference genome. So you might have a human genome and you compare with a whole bunch of mammals. And of course, what you normally would see that lines to that would be what's found in the human genome. And of course, if a piece of DNA, a sequence is not present in that human genome, what do you do with it? At the moment, you kind of blip it out. But there are ways to visualize that where it kind of pops out. So there are various tools for looking at that. So I think maybe that's one of the routes that things might go. The other point I wanted to raise was in this audience, there are a lot of people that have actually worked together for probably a long time, 15, 20 years, in genome projects. So there is some relationships already, which I hope will kind of build on in the FANG thing. I don't know, people, probably, I know a lot of people around here, probably at least half the audience in here have got some connections with one of those kind of projects. So hopefully the rest of them will see. It's actually good fun to share a lot of things together. You do get a lot out of it.
Paul, if I could ask a question. So I appreciate your point about that it's difficult to get money because you're invisible and you only become visible when there are problems. But you indicated also that you were successful in gaining money to stay invisible. So could you tell us what your arguments were to get funding?
Well, so there's actually a number of them, and partly, of course, you know, we have a block of money. About half of the funding at the EBI comes from our core budget from EMBL. For those of you who don't really know the structure, the European Molecular Biology Laboratory is a treaty organization where 21 countries who have signed the treaty pay money into a pot. This pot gets distributed between the 5 sites of the European Molecular Biology Laboratory. The EBI is one of those sites. About half of our budget comes from that source. The other half comes from competitive grants, where we have gone and applied to the Wellcome Trust, the European Union, the NIH, and received competitive grants. The arguments we tend to make are about creating value in these resources. We tend to make arguments about the difference in cost from generating data to providing it out again, and the labor saved when you organize things in a coherent way. We have been comfortable using, say, large-scale compute to do analyses that we can then pass on to other people, because if everyone had to do some various task, that would be, it is inefficient from the point of view of just time spent. One argument we haven't made yet, but I think we should make more strongly, is arguments around reproducibility in science. Most of you who have ever interacted with a bioinformatician know that if you take 10 bioinformaticians and give them the same RNA-seq data set, you will get 10 different sets of enriched genes. This doesn't actually help very much. And if we can remove some of these variabilities that exist for technical rather than biological reasons, we make advantages. So those things are important, but obviously to keep making these justifications, we have to demonstrate usage of the resources. If all of the data, for example, in the 1,000 Genomes Project, was downloaded 3 times, it would be very difficult for us to make an argument to continue to receive funding to support those resources. Now, that wasn't the case for the 1,000 genomes, but there are other projects that necessarily have a lifetime that ends because of usage and demand and things like that. So, and we do, although maybe not as often as we should, we do close things and we do shut down some of our resources. We tend to find people use them in very weird ways when we turn them off, ways that we didn't expect. Again, that's a good thing, but it's often useful to move people to new things, updated versions, and so on. So I hope that was, it's a complex series of arguments, and different arguments work in different situations.
Thank you.
So, Paul, I'm going to change the direction a little bit and go back to your own research that you have with Duncan.
Mm-hmm.
So, as we bring on new genomes, whether it's cattle, multiple bird species, go down to decapods and others, I think that that's probably one of the places where those new tools and that uniqueness will come. But in what you've done in your own research, what did you learn to say, okay, what are the pieces that we need to find and how to solve these questions to utilize that richness of data that should come, that hopefully we can build a better edifice instead of just making a few bricks?
So just to give a bit of background, I've known Jim for a while. The research that he's talking about, I've been working with a group at the University of Cambridge, Duncan Odom's group. We do ChIP-seq experiments in the same tissues in many species, and then compare how the transcriptional regulatory networks evolve over time. And so we published a paper earlier this year that looked at 20 different mammalian species, from Tasmanian devils to whales to humans and cattle and so on. What we find from those studies consistently, and what other people have found who've done similar experiments, is that the sites of transcriptional regulation, or where the transcription factors are binding, change very rapidly over evolutionary time. It's unclear what exactly these changes mean in the grand picture. And we get many questions about whether or not these sites are functional. Some of those questions, I think, are a little bit misplaced, in that the functional regions of the genome must be a combination of those that are evolutionarily conserved and those that are lineage specific. It can be no other way, otherwise we would all look like mice. So the question comes now, what fraction is conserved and what fraction is lineage specific, and how much does this matter? And this question, I think, is still very open. And so as we get more functional data in more different species, we have the opportunity to analyze data and especially do comparative genomics in new ways. Most comparative genomics up until today has existed by aligning genomes and looking at how the DNA sequence itself changes. For some people, that's the definition of comparative genomics. I would argue that we have a huge opportunity here. We can do comparative functional genomics, where the function is conserved even if the sequence has changed, and understand where the function has changed when the sequence is conserved, and understand why this dichotomy exists. The mammalian genomes are big and complex. There's lots of non-coding space, and that just creates an opportunity for lots of different, in the way I think about it, regulatory solutions to the same problem. If we do this right, we could discover that various mammalian species are ideal models for, in places we didn't know they were ideal models. We already know that this happens. For some diseases, mice are a near perfect model. For other diseases, they're a terrible model. And rats are a good model, or something else. We have no idea why this really is. We'd wave our hands and we'd say something. But I would argue that that's because of regulatory conservation that currently we don't have enough data to understand. And so the more species we can collect regulatory information in, the more we may be able to better understand where our models are appropriate, are inappropriate, and we may make actually some exciting new discoveries that are directly relevant to places we didn't expect them to be. So that I think is a huge opportunity. That doesn't necessarily need new tools, but it does need to be on the lookout for those opportunities.
So I wanted to make a comment, maybe come back to one concept you mentioned at the beginning of the talk, and it was that it doesn't need to be perfect. And so as we discuss new annotations or new assemblies or including variation in the analysis, I wonder, I mean, to what level do we need to really try to include every single bit of information, or is there a need also to make an effort and identify, okay, for these species, what are the regions that we can trust, that we can analyze those? Maybe it's 80%, 90%, 60%, but at least we can make more reproducible findings. And so in your work, comparing across species, do you do any of that? Do you identify like these blacklist regions, or how do you deal with that, or how should we deal with that?
Yeah, so these things are necessarily complex. We do a few things. I don't think we have this problem solved by any stretch of the imagination. Like, we spent a long time in this 20 mammals. The quality of the genomes varies dramatically. We didn't always have the genome of the right species, but some near species. Analyzing how to make the ChIP-seq data, in our case, comparable, which was a challenge. I think we ended up subdividing our results into results across the big 20 set and results across a smaller set of 10 species where we had better alignments. But I think that things generally, they're generally applicable in both situations. But this is an area to basically to try to face the complexity. We will get better genome assemblies, that will help, but it won't solve all of the problems. And so to try to, I think you're right, to try to identify places where we are likely to be more correct is critical. But we shouldn't be too worried that we will make mistakes. And one of the, to me, there's a flip side of an open data issue here too. If we hold back lots of data and then release things that are imperfect, we open ourselves to greater criticism than if we release things early and say, look, this is imperfect, but we want to get it out there as quickly as we can. Some of you may remember when the 1000 Genomes Project first released their SNP set 2 years before their first publication. Many people stood up and said, oh, I told you the 1000 Genomes Project was a giant waste of money. Look, they have errors in it. At that point, no one had ever done an analysis like that. And so it was imperfect. Much better now. And I think the process of putting things out there for other people to look at and criticize, and to accept that criticism as graciously as possible, which isn't always easy, is an important part of figuring out how to manage the imperfects of what we will do.
A couple of comments. One on the quality of the reference genomes. The new assemblies that will be out in the next 6 months or so are going to be probably 2 orders of magnitude better contiguity than the current ones. So it's going to be a spectacular improvement. And just to draw people's attention to the fact that the paper Paul was talking about, the 20 odd genomes in that, and the 10 better ones that were the focus of the more detailed studies, in that top 10 were pig and cattle.
That's right.
The other comment is a somewhat disturbing one. I was just checking how we were doing on social media against the hashtag FANG and came across this entry from August the 21st. The FANG has lost over $90 billion this week. FANG in this context, apparently, and this is really disturbing, Paul, includes Facebook, Apple, Amazon, Netflix, and Google.
So there is a funding opportunity here that we have not yet discovered. So if Apple is listening, I'm happy to wear an Apple T-shirt for every talk in the future, if that works for you.
So when you were discussing infrastructure development, you were discussing it in the context of specific projects. The Human Genome Project, the 1,000 Genomes Project, the ENCODE project, all of which had a very well-defined scope, a beginning, and an end that may or may well not have been adhered to. People, and all of those were set up to work with the data generation centers that were approved and blessed. People are dutifully depositing data in places like the Sequence Read Archive and other places. If we can get the thorny issues associated with metadata that we seem to always get ourselves wrapped around the axle on, would these infrastructures be scalable to absorb all of this other data that's being generated by all of these other funding mechanisms and use them productively as parts of those projects?
So the infrastructures, I'm confident to say, are scalable. This is our entire reason for existing at the EBI. It's also the entire reason for existing at the NCBI. And that was one of the points of showing that our data archive growth curves. When next generation sequencing data first started coming in 2008, pretty much everyone who had an opinion, which is pretty much everyone, told us that it would be impossible for us to store this data. Not just impossible, but stupid and irresponsible, and many other adjectives that I can't repeat up here. Those people turned out to be wrong, actually. And it wasn't obvious, although we were confident, it wasn't obvious that we were right. And that it's possible to store these data and continue to scale. So we have benefited from being tested by what was a gigantic step change in data coming in that happened in 2008 and 2009. We have benefited from the fact that Google and Facebook and all of these other companies are also driving down the cost of computer infrastructure. So those things are true. For the question about older data sets with incomplete metadata. Yeah, we can handle the new stuff. I think we should take a look at the old stuff, save what we can, but not work too hard at it. And if there are things that are obviously salvageable, great. But in all of these things, the new data so quickly overwhelms the old data that you never have to delete old data. Like, none of you ever delete any of your old email, right? You just buy a bigger hard drive because it's too hard to get rid of old stuff. And the new data always represents, data from the last 3 years, based on the doubling times we have, always represents 85% of our data resources. So everything more than 3 years ago is only 15%. So yes, I think we can scale this. Doing it in a way that when we go back in 2020 to pull up, to grab a piece of data that was generated in 2015 and have it be useful for that question we're finally pulling together, it's easier said than done. And so we do have to be vigilant on that.
Yes, if I remember correctly, the South Genome Project now have over 2,400 individual genome has been analyzed. I guess we all understand the number matters, but do you think the 1,000 is enough for the 2,400 is better, or is there a better way to determine how many individuals and numbers we need to analyze?
Yes, the 1,000 Genomes Project ended up analyzing 2,504 genomes. This is a number that I know well. We did this because it was possible, and the project was named. So I can tell you at the start, a lot of people didn't believe it was even possible to get to 1,000, and the name was partly put in place because 1,000 is a big number. And 1,000 is also an evocative number. When people say things like a picture tells 1,000 words, they don't mean exactly 1,000, they mean a lot, and people sort of understand, I hope. So what I think, in general, should set an ambition that is as big as feasible, and as big as kind of imaginable that could actually be done. but then take the opportunity to grow it if that happens. And it becomes, because of cost and infrastructure and other things, possible. So the 1,000 Genomes Project also had a lot of delays. It was originally supposed to be a 3-year project, and it took 8. Partly that was getting samples, partly that was doing analysis.
Really interesting from the time we made the first data freeze for a particular paper, the publication of the paper was always at least so I'll start again for the mic. From the time, for 1,000 genomes, from the time you made the final data freeze of the sequence data, including the alignments, to the time the paper came out was generally 2 years. And that's where the project took 8 years rather than 3.
Yeah. So the final release, which just came out, well, the papers came out, but the final release is dated the 5th, or the final freeze. Yes. Right. So we froze the sequence in May 2013. The variants were finished and released to the community in the summer of 2014, and the paper was published in October 2015. And that's the situation where the paper is a good thing to come out, but all of the variants were already widely in use in the community for 15 months before that. And that's, when I talk about the scientific high ground, the 1,000 Genomes Project did a good job of putting its data out there in that way.
And I just wanted to add to your comments very briefly. Maybe this is a bit redundant and people already know it. But the 2 issues that I think are important is that you have a period where you're gathering data where standards evolve and how you handle the data evolves. And then you set a freeze. And though you might have several freezes as methods improve before you release. So don't think that you have to get it all right on day 1. And I just think that's worth saying. And the second issue is that if you're going to rely on the public repositories to provide this foundation infrastructure, then you really need to get them heavily involved as partners at the beginning. Otherwise, they will be dictating to you when those data freezes are, rather than you working with them. So just don't assume that activity is already funded for you.
So the process that we have established at the EBI, which is not exactly the same process as works at the NCBI, is we have sought funding for what I would describe as the project-specific fitting. And so how we engage directly with the project, and then on the other side, engage with the archives at the EBI, rather than funding for the archive itself. This has worked for us, but you're exactly right. Large-scale projects and large-scale projects with various complexities require consideration all the way down the informatics stack.
Please, thank you very much for the lively discussion. Please join me in thanking Paul one more time. Thank you to everyone for participating so well so far. I have one quick set of announcements before the break.
Actually, just to tell you that it's break now. But the main point is we have a very full hour from 11:15 to 12:15. So we'll warn you with a few minutes to spare, but please be in your seats at 11:15. Thanks.